Okay, great. That should be it. There we go. And then we just want to share the screen.
You just have to unmute just one.
And then in the full screen mode.
Okay. So I did hit the unmute button. Can you hear?
Oh, I couldn't a minute ago, so I'll go check again.
Okay.
Okay.
Okay. Hopefully that should be it.
Okay.
Okay, great.
Cut off too much of the phone.
Or you can hide the floating controls.
Yeah, I don't know how that
It's under
disappears.
more. And it says hide floating control, reading controls.
High floating.
Yeah, I can find
Oh,
it.
fantastic.
That's my one trick.
Fantastic.
This changes my life.
Okay, great, I know. Wonderful, well, thank you so much. So, a huge welcome to everybody who's here and everybody online. Thank you so much for coming to the next Chairs Lecture Series. It is a pleasure to introduce Greg for the seminar today. And I have a few words I'd like to say. Just a quick note before we get started. So the next lecture will be given by Debbie McKenzie, and that'll be on February the 9th. And at that time, we weren't able to do it for today, but we sent out a survey and we had about 50 responses that came back with regard to having it hybrid, in person, etc. The vast majority of respondents said that they would like to have the Chairs Lecture Series hybrid. So we will continue doing that as we have today. But in addition to that, the majority, about 75%, asked to bring back the food and the social atmosphere and that aspect of it. We weren't able to get it ready for today. We will from the next Chairs Lecture onwards. So from February onwards, there will be some snacks and some drinks and that type of thing here as well. So, okay, great. So as we get started here, so it's a pleasure to introduce Greg. Just a few words here. So Greg so he has a broad research program spanning from environmental physiology of animals to environmental toxicology with a particular focus on fishes. Greg has published over 230 peer-reviewed journal research articles, plus 1 book and 4 patent applications. So he's been extremely productive in his career. He completed his PhD at the University of Ottawa in 1993 with Dr. Steve Perry and had 2 PDFs, one CIHR funded in cell biology at the Hospital for Sick Children in Toronto, and one at the Heart and Stroke Foundation funded in molecular medicine at Harvard Medical School. Greg started as an assistant professor here in the Department of Biological Sciences and has risen through the ranks to become a full professor in 2006. Greg has received numerous awards, including the Petro-Canada Innovator Award, the CSZ New Investigator Award, American Physiological Society Young Investigator Award, a Killam Annual Professorship, as well as a McCullough and the Faculty of Science Research Award. So again, very productive and very successful. He has trained 34 PDFs, 20 PhD students, 14 masters, and over 100 undergrads since starting, with more than 20 of his former trainees in academic positions worldwide. In the toxicological sciences, Greg works broadly with industry, governments, and academia to examine the environmental issues related to water sustainability and regulatory science. His research includes studying the effects of various biotic and abiotic stressors and pollutants on aquatic animals. Micropollutants studied include nanomaterials, pharmaceuticals, and personal care products, hydrocarbon contaminated fluids, and the micro and nanoplastics. He has served on multiple local, national, and international boards and advisory panels. Finally, Greg has had an annual continuing program at Bamfield since 1999, where he investigates the physiology of marine fishes, primarily dogfish, ratfish, and hagfish, and has published over 30 papers on hagfish physiology since 2014. So the seminar today is on his hagfish work, and I'm excited to see what Greg is going to tell us about today. So Greg, it's all yours.
Well, as I mentioned before, the people who were here, PowerPoint updated this morning at 11, and it's been giving me grief ever since the last 3 hours. So hopefully we don't end up with any grief. First of all, thank you everybody for attending, and those attending online, I appreciate it. And thank you, Declan, for the opportunity to give a talk. I normally don't get to attend these because I teach on Thursday afternoons, which makes it a little difficult if it's always on Thursdays. But I'm going to talk to you today about one of my favorite little critters, and that is the hagfish. So, and the work that we've done out at Banfield for the past 23 years or so on this little critter. So if we go to, hopefully this works, I have to maybe get a little closer, and it's not working, so get a little closer. Did that work? If not, we'll use this. Okay, PowerPoint, tell me what you're doing. Okay, that's only with this mouse. I'll have to use that one. So Linnaeus originally described hagfish in the annelids actually as a worm, and then he quickly corrected himself a couple years later. And the original definition of hagfish, essentially, if you look in the 1758 book that he has, it enters into and it eats fishes, changes water into slime. And I think that's what people know much about hagfish. And we'll come back to that because the first part, enters into and eats fishes, is an important component. So this is what people normally associate with hagfish. This poor individual got hit from behind by a truck carrying hagfish, multiple buckets of hagfish. And you can see this, my email blew up when this happened because 1 million people sent me emails asking me about it. But I think we're all familiar with slime production. They can produce copious amounts of slime. Literally, a 5-gallon bucket can be turned into a full bucket of slime within a few seconds by handling the animal. And that's a 100-gram animal. It's incredible. And so you can actually, it's thick enough, you can pick it up. And this is a defense mechanism that hagfish have. So if they get bit by a shark or a sea lion, or etc., it immediately throws a bunch of slime into their mouth. It chokes off the gills, and they spit it out. And it's an interesting defense mechanism that they use. But it's only a face that only a mother could love, right? It's not a species like these large charismatic megafauna like elk or like wolves. We have all these fantastically beautiful species, and they're charismatic, but I think hagfish are charismatically ugly. Right? They are a special animal in the vertebrate world because they are what we think are one of the most basal vertebrates present. And we'll go into there. Koreans love to eat them. About 50,000 tons of hagfish per year are eaten in Korea. And they literally barbecue them alive. So animal care, right? So this is a fairly famous dish associated with Korea. So, and also they're used in eel skin wallets. Their hide is tanned. If you have an eel skin wallet, it's actually a hagfish skin, right? And so that's what they use. They make it into a leather, and that leather is very effective as a nice wallet. Before I get going, I think it's always important to tell about the people thank the people who actually do most of the work. Now, the reason I go to Banfield is I get to be a scientist at some point instead of somebody just pushing papers. What is happening? Thank you.
Went too far.
Yeah, that's the problem with roll tops. So the, going to Banfield, I get to actually do work in the lab, inject animals and do surgeries and all the other things. So it's a wonderful experience. But the people who do the work are, some of them that I'm going to talk about today are here. So Alex Clifford, a former PhD student who's now down in San Diego with my first PhD student, Martin Tresgueres. Martin and Scott Parks, Martin Tresgueres and Scott Parks started off our work in hagfish way back 15 odd years ago. I have a very strong collaborator in Sue Edwards, who's now the president of University of Dayton. And, we get together in Banfield, but you can see there's a large group of people that I call Team Hagfish, which are the ones that out in Banfield have been able to work, and we've collaborated with a large number of people over the last 15 years working on hagfish. So the interesting part about hagfish, gonna go very slowly here, because the interesting part about hagfish is its placement in the evolutionary tree has been quite controversial over the past 30 years, but more recently been fairly well resolved, although there's still some questions that remain to be answered. So for about the first 90 or 100 years that people started putting these into phylogenetic lineages, it was thought that hagfish formed a monophyletic lineage separate and distinct from lampreys. Right? This is the idea. These are the agnathans, and it's the most basal vertebrate present. And so that was mostly on morphology, so the evolution and morphological components as related to their mouth parts and a few other components. But more recently, and I think it's been resolved very effectively, first by a paper by Heinberg, where she looked at RNAi, microRNA, and where he showed that, or he suggested that hagfish and lamprey share a common ancestor, so it's a monophyletic lineages of the two components. And a real talented person who came out of this department, worked with Phil Curry and also worked with Rich Palmer, Tetsuda Miyashita is now at University of Chicago, and he pretty well put the nail in the coffin, saying it is a monophyletic lineage shared with a common ancestor between hagfish and lampreys. So this creates some really interesting questions because they're quite different animals. So what's different? So my PhD and master's and most of my postdocs were all working on ion regulatory physiology, how animals regulate ions and acid-base status. And so when I was looking for a program to go out to Bamfield, so I could go out to Bamfield because that's what you have to do, right? I was looking at hagfish, and they're quite unique and interesting because unlike lamprey, which they have their ion concentrations far lower than that in the seawater. So here we see sodium being 439 millimolar, chloride being 513. That's in the seawater. And you can see that all these other animals, almost all the teleosts, the sharks, lamprey, they all have much lower sodium and chloride than the surrounding water. But hagfish are more like invertebrates, because invertebrates are ionoconformers, and their ion concentrations are almost the exact same as seawater. They're the only vertebrate that's like this. Right? I wish this thing worked, but I guess not. So Homer Smith in 1932 described this. He said that hagfish were the only vertebrate osmoconformers, and we haven't found anyone since. Crabs, etc., they're all ionoconformers, all the inverts, but this is the only one that's a vertebrate. So that's pretty unique. It's a strategy that's kind of strange. And remember, they're sharing a common ancestor with the lamprey. They do maintain their plasma compensation similar to seawater. They do regulate. So when people say they don't ionoregulate, that's not true. They regulate their divalent cations, calcium, magnesium sulfate, because they have adverse effects if they're on physiology, if they're dysregulated. So they do regulate those. If they get too high, they cause problems, specifically with phosphate mechanisms, because they'll precipitate phosphates. And the question is that now an ancestral character, or is it a derived character? Because if you share a common ancestor, your ionoregulatory strategy has now reverted back to one that's similar to an invert, or is it, and the lamprey where the common ancestor had the ionoregulatory capacity? So those questions are quite unique and interesting. So it really creates a fantastic question in biology about how do we get to this ionoregulatory strategy. Now, I'm not going to talk a lot about, I'm going to focus, because we've done, like I said, over 30 papers in the last 8 years just on hagfish physiology. But I'm going to very quickly show you that they are incredible extremophiles in that they are champions of acid-base regulation. They can take acidifications far better than almost any vertebrate that we know. They can handle CO2 better than any vertebrate that we know. They can handle ammonia, and that's what I'm going to talk to you about today, the physiology of how they handle ammonia, which is incredibly toxic. They can handle it. They're the only vertebrate to be shown to use their skin and the epithelium, external epithelia, to actually acquire nutrients like phosphate, alanine, amino acids, etc., directly from the water. They don't need to eat. They can just acquire it. As long as it's present in the water at high enough concentration, they can take it out, which is really neat, and no other vertebrate does this. So you can see there's so many different things that you can get excited about in terms of hagfish physiology. So what are hagfish? This is the prototypical idea of what's happening with the hagfish. Remember, they're agnathans. They don't have jaws. These are not jaws. These are keratinized epithelium. So it's kind of like a tooth, but it's a dermal plate as opposed to a jaw, a true jaw. So they're agnathans. They are voracious feeders on and demersal scavengers. If there's a whale fall or a dead a vertebrate or a fish that falls to the bottom of the ocean, they are there in minutes. They will be the first to arrive. They can come from kilometers away. And that's because they have really fantastic sensory organs on these barbels, so they can detect directionally where that food is and come, but they're within minutes on top of an animal. They're the first ones there before the crabs, before anything else when that whale falls, as an example. So this is a prototypical example. This is actually a picture from off Monterey, I believe, of a hagfish on a carcass entering into it. And you can see they're sort of facing into that carcass. And this becomes important as we see later on. So they tend to be curled up and wait there for months sometimes, and then they'll just go and eat. So they have to eat when they have a chance, because it's a very nutrient poor environment. And when they have a chance, they get it as quick as they can, because that's what's needed for their growth and reproduction. They do actively hunt things like fish. There's videos of them actually hunting fish and going into tube worms, for example, and it's the long skinny, so they'll go into the big tube worms and eat the tube worms. So there are a variety of ways that they can acquire food, but generally they're just sit and wait for scavengers. Well, that worked great. So I'm not going to tell you much about this, but as an example, this is for acid-base regulation. I don't know if I can get rid of that because it'll just come back as well.
I think it's only coming back because Linda keeps admitting people.
Okay.
She restarts the power.
Okay, it doesn't matter. It just says extraordinary acid-base regulation. So normal pH for a fish at 15 degrees, 14 degrees, whichever, is around 7.8 for their blood. We can actually acidify their plasma all the way down to pH 6, and they can bring it back in a few hours. If you take a normal fish, you could go to about 7, or else they die. And over maybe 48 hours, they'll bring their pH back to normal. They are bringing their pH back to normal within hours, because they have an incredible capacity to acid-base regulate. So they can have repeated infusions of a dilute buffered hydrochloric acid solution or sodium bicarbonate, and they can really ramp up their ability to acid-base regulate. This is another one in terms of base regulation of CO2 tolerance. So CO2 in mammalian tissues like ourselves are around 40 torr, 5% CO2. That's why we use it in the incubators at 5% CO2. So that's around 35 to 40 torr, so around in this range. Normal vertebrates, or all vertebrates, can only raise their bicarbonate levels to compensate for an acid-base disturbance to around 30 millimolar, because that's the ratio between CO2 and bicarbonate that gives the proper pH. Hagfish can go up to 80, even 100 millimolar, 3 to 4 times higher their plasma bicarbonate than any other vertebrate species. And this is, and I think actually my hypothesis is that this is the reason why they reverted back to that ionoconforming strategy, because by having a higher chloride concentration, they can dump chloride and retain bicarbonate so that they can acid-base regulate, and other animals can't. That's something that we're trying to follow up on. So you can see that if you take a normal animal that's at, say, 2 torr in the water, right, and you put it in higher CO2, this is the increasing concentrations of CO2, they raise their plasma bicarbonate up so that their pH comes back close to normal. And that's how you read this really complicated, what's called a Davenport diagram. So I'm not going to talk about acid-base regulation. I've had some fantastic postdocs and students working on that. Scott Parks, Marina Giacomin, etc. But I'm going to talk more about the scavenging lifestyle and some unique components about that scavenging lifestyle. So remember, Linnea said enters into and eats fishes. And not only eats fishes, they eat everything that falls to the bottom, including whales, seals, sea lions, etc. And so inside those animals, when they die, it gets pretty grim, right? If you think about what's happening, there's going to be, when that animal is died, they need to acquire nutrients, but there's also going to be high partial pressure of CO2, high ammonia concentrations, low oxygen concentration. So if they enter into a dead whale or a dead sea lion or a dead seal, inside there, all the bacteria are putrefying, it's getting disgusting, right? They're consuming oxygen, they're releasing all these nutrients, and this animal goes inside there and has a grand old time eating away while other animals can't get in there because it's too toxic for them. So one of the earlier papers by Chris Glover and my former master supervisor, Chris Wood. Chris Glover is here as a from Athabasca University now, but they worked out in Banfield, and they showed that the skin could actually take up amino acids as a part of a study. So we actually went to continue with that a little bit. And we showed that phosphate, which is an incredibly limiting nutrient, especially in the ocean, it's at between 0 and 4 micromolar. So all the high calcium that's in seawater immediately precipitates with calcium and causes and removes the free phosphate, and that's something you need for growth. If they're inside that soup and there's free phosphate, they can take it up really quick. Right? So they have to do it as fast as possible and get it so that then the animal is going to be stripped clean soon by all the other animals. So they really have trying to take advantage of this unique lifestyle. So it got us thinking, what's it really like inside a dead animal? Now, couldn't really get permission to kill a whale and drop it. So what we did was we started looking at different ways that we could imagine what kind of putrefaction. And we took our hints from the idea that there's a whole science of forensic science that looks at how long since somebody has died or an animal has died. And inside that, you get an early stage decomposition, so initiated by bacteria in the gastrointestinal and respiratory systems. They start going crazy because the immune system is not functioning anymore, and of course they can now start to multiply. And they generate hydrogen sulfide at really high concentrations, lactic acid, propionic acid, butyric acid, which is what really smells bad, methane, and lots of ammonia. And in the soils around, say, a dead carcass, you can get like 29 millimoles per kilogram, but that doesn't tell us much about what's happening inside an animal in water, right? But we've seen pictures like this. And so this is a video of hagfish. They're feeding on a whale. You can see they're sort of sticking their mouth on the surface, and they're going to pull off, and they're going to go through a hole and into that animal. If an animal putrefies, this is obviously an extreme example of a fin whale that was putrefying. This is all blown up because all those gases are building up. And if you search on YouTube, you can find pretty gross videos of people puncturing this and getting covered in absolutely disgusting stuff. So this is a fish that we actually fed to a single hagfish just to see what was happening. And remember, I said it enters into and eats fishes. It went in here and it hung out inside here, and it goes for the liver first, because that's the high nutrition part of the fish first. So hagfish, and this is them just going crazy on a fish at the bottom. It's another example. So you can see the hagfish are trying to dig into that carcass and get in there. So a paper that we just published a few months ago called with Alexander Clifford, my former PhD student, with some colleagues, Mike Wilkie from Laurier University, Susan Edwards. At that point, she was at Appalachia State. Martin Trezier, my former PhD student, who's now at Scripps as a full professor. He just made full professor, and myself did this study where we looked at the ammonia tolerance, because if you think about going inside that dead animal, there's really high ammonia. So what we did was we wanted to see what it would look like. So we took literally falcon tubes with a piece of liver or a piece of meat or muscle, and we put it in the seawater at the right temperature, but we sealed it up and just we put a little bit of bacteria from the intestine in there and just let it build up for days. And then we measured inside, monitored what the concentration were of ammonia or the pH of the water or the PCO2. We also monitored oxygen. So oxygen disappears within a few hours. If that minutes, like 50, 30 minutes, and oxygen was mostly gone because it's sealed. The water pH dropped because of all the acids, propionic acid, butyric acids, etc., to around pH 4. Right? They're hanging out in pH 4 water, no problem whatsoever. The PCO2, which is remember I told you that 40 torr is about what is normal for inside a whale or an animal or a human or whatever. And so the CO2 can actually get up to like 300 torr. So about 9%, 10% CO2. So incredibly high CO2, and CO2 goes in and will cause acidification inside the animal. So high CO2 and low buffering capacity creates that issue. But I'm going to point out today mostly that we're going to talk about this ammonia. So this is the time in days, and our control had no ammonia generated because we didn't have any tissue in there. And then if you look at the amount of ammonia that's building up over the next few days, it gets 90, 100 millimolar. And you think, well, what is that? Is that really a problem? Yes, it is. I can tell you for almost all vertebrates. So ammonia, I'm going to see if I can get rid of this trick here. No, Linda, if you're listening, can you do the more and get rid of the floating, what's it called? Floating bar, whatever.
Floating meeting controls.
Floating meeting controls. So nitrogen toxicity, and nitrogen and ammonia is really super toxic to animals. There's a buildup of ammonia that causes overexcitation of the immune system, of the nervous system. Activation of NMDA receptors because it substitutes generations of lots of reactive oxygen species, causes eventual necrosis and apoptosis. This has been studied very extensively in neurobiology, ATP deletion and encephalopathy. And the toxic range for you, for I, or for almost any fish, maximizes around 0.5 millimolar. Remember, I just showed you a graph that had 100 millimolar ammonia. And these fish crawl inside and hang out. So now there's a soup, and and their skin is acquiring phosphate, their skin is acquiring amino acids, their gills on the outside are acquiring it, they're eating, they're trying to gather it as much as possible. There's no oxygen, there's toxic levels of ammonia, there's toxic levels of CO2, and the pH is incredibly low. They're I think the best extremophiles that you can imagine. So you have this really neat animal that does this really neat stuff. So let's study it. How does it do it? So for nitrogen, it's a little complicated because for ammonia, you have ammonia and ammonium in equilibrium depending on the pH between those two components. Right? And ammonia, for almost all fishes, what they do is because they have lots of water around them, they just ammonia will just diffuse out into the water. Right, so it's got high water solubility, high lipid permeability. This is, questionable now because I'm going to point out that the RH proteins are actual transporters for ammonia and ammonium. There's no energetic cost to excrete it, and there's lots of transport pathways. So some animals, like all fishes, except for a couple very specialized ones, excrete as ammonia because it's cheap and easy. You and I and sharks convert that ammonia from a metabolic waste product into urea. Animals like birds convert it into uric acid, right? Because they have different amounts of water that's needed, and there's very specific reasons for that. So if an animal's challenged with high ammonia, they can convert it into something far less toxic, say uric acid, which has very low toxicity, or moderate toxicity like urea, and that leaves nest water. But ammonia is very toxic. So physiologists like to draw models, cartoon type models. It helps us visualize what's going on. So I'm going to bring up the fact that RH glycoproteins, your RH factor in your red cell is actually an ammonium transporter. That's what it is. Your RH positive, RH negative, that's an ammonium transporter that was defined in about 99 in the science paper. So rhesus glycoproteins are ammonium transporters. And really, they're just channels that if you have a partial pressure of ammonia gas, it can move through an RH channel. And there's a number of different isoforms. There's some that are basolateral and some that are apical, right? And if you have a high partial pressure of ammonia, it just diffuses through those RH channels and gets rid of out into the water, and it's passed and you don't have to worry about it anymore. Right? Ammonium, on the other hand, can act like a sodium or potassium ion and be transported at very low rates, but in general, ions don't cross membranes, only gases do. So now we have a system where, so if ammonia gas moves, because of the equilibrium constant, you essentially convert ammonium to ammonia, get a proton, and you just literally replace that ammonia and it keeps going. So as it's moving through, you can just get rid of ammonia very quickly. The question becomes, what do you do when you have high external ammonia? Because RH glycoproteins are bidirectional. If I put ammonia outside this animal, so if it goes into that dead carcass, and you have high ammonia, these are transport channels that actually move in the opposite direction, right? Because it's just bidirectional, it just works on the gradient, it's not active, right? And so that creates a bit of a problem, because as soon as you go into a high ammonia environment, ammonia rushes into the animal and it dies, right? But not hagfish. And so you have these apical and basolateral transporters. So that's essentially what will happen if you go into a high ammonium environment. So you can convert to less toxic forms, as I told you. Hagfish don't have the enzymes for it. They're before all the enzymes for the urea synthesis cycle are missing in hagfish. They don't have it through their genomic rearrangements. There are some animals that do have some fish that do produce urea, like toadfish and lungfish, but hagfish do not do this. Some animals have been suggested to volatilize ammonia, so the weather loach, which is another Southeast Asia species, or the mud skipper, again in Southeast Asia in Singapore. They suggested that there was some active ammonium excretion, but they didn't really go into anything mechanistic for it. So our hypothesis or our idea was that hagfish must be able to tolerate, detoxify, or eliminate ammonia. And when I say ammonia and ammonium, right? When you experience going into that putrefying carcass. So what we did was we simulated this in a typical bucket experiment, physiologists in the field. We used buckets, put fish in there, 5 or 6 or multiple replicates of fish in there. We'll put them into high environmental ammonia. So we did about 20, so it's about 1/5 of the maximum value, but one that we think was reasonable. And then we recovered them, and we can measure the rate of ammonia going in and out of the animal as a understanding of what's going on. And this is Sue Edwards and Alex doing some work at Banfield. And so we can do a variety of different stuff with them. And so what I'm going to show you here is where almost every fish and almost every vertebrate, with a few exceptions, the one that I just mentioned, will die if you get above 500 micromoles per liter of ammonia. They won't make it. So if we put 20 millimolar ammonia into that animal, or into the water, sorry, what happens? Within 4 hours, the ammonia is up over 2,000 micromolar. So within 4 hours, any other fish that goes into that will be dead. But it keeps going up and up and up, and it gets up to 5,000 or 6,000. This is the highest plasma ammonia ever recorded, by far, by 3 times, 4 times. Right? So they are incredible species, because not only do they have a capacity to withstand it, they actually have a capacity to excrete it actively. And that's what I'm going to try and prove to you now, and what we came to the conclusion on our paper that we just published in Active Physiological. So they are extremely ammonia tolerant. Right? So I'm going to show you this slide here, which is a little complicated, but you should be able to follow, I hope. So what we did was we varied the amount of environmental ammonia. So we put in 1 millimolar, 2 millimolar, 5, 10, 20. And then we looked at how much was going into the plasma. So this line is actually the line of equilibrium. You would expect as if it was purely passive, that as you had a higher and higher ammonia concentration, you would get higher and higher plasma. So we expected all these values to follow along this line. But when we did this experiment, we did it multiple times, depending on the group that we had, you can see that always the plasma ammonia was far below the environmental ammonia. So for some reason, they're able to kick out ammonia. But remember, ammonia is a gas that goes through channels. It's also an ion. So how are they doing this? How are they getting rid of ammonia and being able to go into that really toxic environment and survive? So we think that this, 5,000 to 6,000 micromolar is the limit of their toxicity. We didn't go above that, but it's pretty well what we call a ratio of 4 to 1 over the environmental ammonia is 4 times higher than the plasma ammonia. And so you can actually measure if you know the concentration and your time, you can measure the plasma ammonia. So that's shown here, right? And we can actually look at the flux or what's the rate of ammonia. So when we first put them in, that ammonia comes flying into the animal. So by plasma ammonia, you can actually calculate the rate of ammonia uptake. And you can see that it's going into the animal quite readily. But very quickly, within about 16 hours, somewhere between 8 and 16 hours, they switch over to being able to excrete that ammonia and reduce it. And they maintain that throughout the entire period. So they're actually going to excrete this ammonia fairly quickly. So what I have over here is what we call it my hagfish gas gauge. Right? And essentially it looks at the fluxes or the ratio of how animals or how hagfishes respond. So this is one of the bugs that came out in the new version of PowerPoint at 11 o'clock in the morning, right? If you raise the water ammonia concentration, it directs an inward gradient. So now what that causes is the plasma to go up in a ratio of about 4 to 1, right? So that's what we expect ammonia is transmitting. But then if we go over time, they can now excrete the ammonia against a concentration gradient. So if you know your transport physiology, going against a concentration gradient requires ATP, requires it's an active transport mechanism. It's no longer passive. Right? So they can resume it very quickly and increase that. And because it takes a little while to do it, we think that there's an upregulation of the capacity, right? And so we wanted to figure out how. So what we could do is go back to our model and say, when we increase the amount of ammonia, we reduce the amount that's coming back in, or we increase the value going out, and that comes into an equilibrium. The rate going in and the rate going out are in equilibrium, that's where you'll sit. Now, going back a ways, right? August Crowe, who won the Nobel Prize in 1939 for physiology and medicine, he proposed for fish that there's a linkage between sodium uptake and ammonium excretion. This was in his book in 1937. And for about 35 years, 40 years, people decided that that wasn't proper. And so then they started saying it was only protons in ammonia. So that's the sodium proton exchanger that is a very significant acid-base regulating protein within all cells, right? And the idea was that sodium proton exchanger serves as a mechanism. This is what we tried to figure out. Is this the mechanism? Because NHEs are what I studied when I was at Harvard Medical School for my research project. So remember I told you that ammonium and ammonia come into equilibrium with a pK of 9.3. That means that almost all the ammonia is present as ammonium and only a small fraction as ammonia. And so if we thought that this transport protein, the sodium proton exchanger, was responsible so that ammonium substituted into this site and was kicking it out, then if we remove the sodium, this should not work, but you'd still have the waste going back. So what we wanted to do was expose to either sodium-free or regular water in the presence that we went to 10 millimolar sodium. So we allowed the upregulation of this transport capacity. Here's our gas gauge, we have our upregulated capacity. And when we do that, under control conditions, the plasma ammonium remains pretty constant within when we transfer it to either sodium-free or artificial seawater. You can see it's pretty constant. And if we remove the sodium that's here, now the plasma ammonium concentration keeps going up and up and up. And we stopped it because we didn't want the animals to die, right? So we only did a short-term experiment. So what that suggests, if you go back to this model, is by removing this sodium, you can't get rid of the ammonia. So the plasma backs up inside here and gets higher and higher. So this is all indirect evidence. It doesn't really give you really good direct evidence of what's happening. So we wanted to figure out a better way to do that. So what we did was we used a chemical called methylammonium, right? Which is a 14 radioactive carbon labeled methylammonium. And so it's got a methyl group instead of one of the protons. And so it's a surrogate as a tracer for ammonium, but not for the gas ammonia. Okay, and so we can use that and say, is it moving as ammonium, the ion? And so we've been looking at the unidirectional flux of ammonia. And what we saw is that under normal conditions, so control and sodium-free, we have very low rates. Remember, hagfish under normal conditions have the lowest metabolic rate of any vertebrate. It's like 1/100th of any other fish. They just sit there and do nothing, right? But as soon as we put them into high ammonia, of course, they have a really high rate of flux. And now we're going up from control of like 5 to 10 micromolar per kilogram to 700. So they can activate this system to really boost and kick out ammonium, or methylammonia in this case, very quickly. If we put it in sodium-free, it reduces it substantially. In other words, we're preventing that from happening. So what we're doing is we're reducing the active transport by removing it. We know that sodium proton exchanger is present because in an earlier paper by Martín Tresgueras, we did sodium proton exchange. This is sodium potassium ATPase and sodium proton exchanger immunohistochemistry under this condition, where we just looked to see whether or not the antibody that we had made actually worked in hagfish. So we said, can we get at doing a really specific experiment where we look at how ammonia moves across from one side of the membrane to the other? And so I know this looks ugly, but this is the inside of a hagfish. So here is our scale bar. So this is 1 centimeter, obviously. All these little white bottles, that's the slime glands, right? You take one of those, put it in the toilet, and it's completely filled with slime. Anfield joke. And exactly. So, but you can see here, these are the gills. So they're about 5 to 6 millimeters big and about 3 to 4 millimeters wide. Each one has a blood vessel going to it, and it has a water duct that goes through it from one side to the other. And so the blood moves in one direction and the water moves in another direction. And I'll show you here a stylized version of what's happening and how we set this up. And this was the first time anybody had been doing this double perfusion protocol. So what happens is you have blood flow coming in under a normal situation. It goes in through the animal, in through the gills, and goes across from the venous side, the arterial side, where it gets oxygenated, and then out through an afferent blood vessel. You also have water that moves in the opposite direction, a counter current exchange. So if the blood or the extracellular fluid has ammonia, similar to what's inside the animal when it's under high ammonia, 4 millimolar. We put this 10 millimolar outside total ammonia. So that means we have recreated that gradient. And so now we have a peristaltic pump, and we do the surgery. It takes us a little while to do this. And we cannulate it so we can perfuse through. So any methyl ammonia, if it is being transported, comes out into the water and appears in the afferent flux ducts. So it's a very complicated but nice set of experiments where we have a blood side, and so the blood flows through and it has methyl ammonia in it, or we have anything that's, if, as the water comes through, it removes that ammonium, methyl ammonia that's been released and brings it in, and we can measure, if we know the flow rate, we can calculate the rate of transport for that material. It can work against the concentration gradient. So here is an example. I'll just, if you can look at the video, you can see we've done a blue dye here just to show you the blood vessels. So we're perfusing this. It's obviously quite small. You can see the blood vessels filling up with the dye. The water is perfusing through in this direction. So we have the blood coming in. We have the perfusing that comes out, and we collect, and we also have the seawater going. So we have it going in both directions. So this is a really neat dual perfused hagfish gill, and we've used this fairly successfully in a number of different projects now. And this is the first publication of this surgical technique to be able to get this. So it's a way that now we have a platform to investigate the cellular mechanisms of what's happening. Right? So there is a drug called amiloride that knocks out sodium proton exchanger. Remember, before I did sodium free, but we can actually use a drug that's specific for sodium proton exchangers and say that it's specific for that particular transporter, because there's lots of sodium dependent transporters on every cell. So we wanted to see if it's specific for that NHE. And so when we did this experiment, under normal situations, we have a flux of ammonia around 1,500 micromoles per gram per minute, which is a fair amount. But as soon as we add the drug that inhibits NHE, it blocks it about 75 or 80%, say 7%, so 60% reduction. So what this, if you look at this model, this is our gill. If we're blocking this, we're preventing now the export of ammonia. So you're reducing your flux of ammonia from inside to outside. Remember, this is happening while there's lots of ammonia outside against the concentration gradient. So you can imagine, you know, like a dam, you have a high value and a low value. How do you get up that hill? Right? You have to have a pump. And this is what dam is doing. It's pumping ammonia. Right? So there is one last problem, the last point that I'm going to bring up. The last problem is the fact that we still have this diffusive gas permeability. So if we have a transporter like the sodium proton exchanger, taking that ammonia and pumping it out against its concentration gradient, so like a dam going up against from below the dam to above the dam, it's pumping it from low concentration to high concentration. Now you have ammonium out here, and it would just go back in directly through the gas channels. So how do you manage that? We looked at the abundance of those RH proteins, and it doesn't change. So none of the treatments. So in other words, they're not turning on new transporters, they're not changing, they're not reducing the amount of RH proteins present in the gill. But we had a hypothesis that maybe they're just removing it from the surface. If you remove that from the surface, then you don't have any back transport or any short circuiting of the ammonia. So the ammonia now can wash away. And so you can't get the ammonia to go back into the animal. So by actively pumping it out, and then you have a small rate of transport going back in, you reach a new equilibrium, which is far below what the outside is, 4-to-1 ratio. And that has to do with thermodynamics of the sodium transporter. Well, I'm not going to get into that for you. So I'm going to blow this up, but this is a figure that came from the paper. So we made antibodies against the RH protein, the sodium proton exchanging the sodium potassium ATPase. And so what I've done is I'm just going to present it in 3 separate slides, so it's a little easier to see. And so here we have immunostatic chemistry showing the sodium potassium ATPase, and we're looking now at the gill surface, but we're looking at the bottom of the cells. So you can, in the confocal, you can move through from the bottom to the top of the cells. So here are the nuclei that are present in these particular cells. These are mostly, there's 2 cell types. One's called an ionocyte, which does all the transport, and one is a respiratory cell, which is just a thin cell. So this is probably pavement cells, respiratory cells. And you can see that on the basolateral surface, right, there's sodium potassium ATPase, and this is a standard marker for the base of cells. The sodium potassium ATPase never exists on the surface. Right? And so we have a nice system here, and if we do our Rh protein antibody, and unfortunately our Rh protein antibody cannot distinguish between the two different isoforms that are present. It's just because they're very similar. But you can see that there's lots of staining on the basolateral surface here, and this is just a standard no primary control that you would have in a metastatic chemistry. So clearly, there's an Rh protein that co-localizes with the sodium potassium ATPase. If we go back to our model, there was that one that was on the basolateral surface. If we do a control animal, one that's not been exposed to high environmental ammonia, so they're not ramping up their system at all. Under that condition, this is the apical surface. So now we still see some sodium potassium ATPase, and the reason is, if you imagine a round cell, the basolateral membrane actually folds up and comes right up underneath the membrane. So it actually has these invaginations on the bottom. So you can still see that. But the interesting part that we didn't see before is on the very surface, when we focus through to the surface, you can see these very intense red staining for another Rh protein. And if you do it, this is a 3D reconstruction of the Z stacks. You can see that each cell has, in the middle, at the very top of the cell, a bunch of Rh protein. So just like we had before when we had the Rh protein potentially short-circuiting us, the idea is it removed from the apical membrane to help in that facilitation. So these are Z stacks. We can look at the Z stack and say where, if it's on the apical surface, you can see it here. This is the apical surface on the bottom. And this is for this particular cell. Each cell, you can see that very intense red staining, the nuclei underneath. If we go to an animal that's been exposed to high environmental ammonia now, and we do that same 3D reconstruction, so I'll just really get you to focus on eye, you can see that Rh protein disappears completely from the membrane. So what it's doing is when it sees high environmental ammonia, it goes into that putrefying dead animal. It's acquiring all the things that it needs, because it only has a short time before all the other animals get there. It's getting all the stuff that it needs, but it's removing the Rh protein from the surface, and it's ramping up its ability to excrete ammonia. So as the ammonia comes in, it gets rid of it. And it has that ratio of 4:1. So, and it prevents the ammonia from coming back by removing the Rh protein from the surface. Really cool fundamental physiology, and really nicely explains how this animal uses an active transport for ammonium. So this is our models. Like I said, this is the published model in the paper. Again, to show under normal low ammonia, we think that we have that apical Rh protein. Ammonia is produced by metabolism. As you produce that ammonia, it's passing through on the blood side, sees an Rh protein on the basolateral surface, and just follows a passive gradient out. When we put the animal into high environmental ammonia, it still has that Rh protein, but now it's ramping up this sodium proton exchanger to get rid of ammonium. Right? And as you get to higher and higher, longer and longer times, we've ramped up the sodium proton exchanger. We've removed this Rh protein into the cellular thing. And now when we get rid of ammonium, it can't come back. And that's the way that these animals can do this. There's only a couple species in the world that have active ammonium transport. And this was a paper, an editorial written by Katie Gilmore from University of Ottawa, who wrote an editorial specifically about our paper. And I just like to point to the last part of what she had. It's a fairly long one, but it seems that time has come to consider the opposite, that nitrogen weighs primarily by diffusion. That is, active transport of this respiratory gas may be important, an idea that at one time would have been dismissed as preposterous. Right? Literally, this is the best physiological evidence for active ammonia excretion. So a summary, very quickly, I'm not going to keep people forever. A summary is that hagfish are ammonia extremophiles, clearly. They're also extremophiles for pH, for CO2. They can go into the most disgusting, horrible places and be the first on and be able to hang out there and feed before any other animal can get there, because it'll kill them. So that's really an amazing feat in terms of being able to adapt to a unique environment. Right? They can stabilize their plasma ammonia. They can still withstand plasma ammonias that would kill anything else, but that's another thing that we don't know how that happens. Right? So we've linked it to environmental sodium. We think there's an active transport. We're actually looking at the sodium proton exchanger, if it can actually function this way. So we've cloned now the sodium proton exchanger. We haven't been able to get the expression started yet. I need graduate students more for that. And obviously it involves internalization of those apical transporters. What this does is it allows these animals to capitalize on carrion drops before anybody else can. They can get in there, they can eat and be happy, and then get out before all the other things.
Is that what's illustrated in that lower right-hand?
This is a
I
whale.
can't quite see what that is.
This is a dead whale, and there's tons of hagfish all over it. Right? The hagfish are the first on the scene. They have an incredible ability to find and to get in and start. They'll go in primarily through, if they can't go through the skin, they often go in through the anus. This is the main spot that they'll go in. Go in through the anus or go in through the mouth and then start eating from the inside out. But remember, they have skin that can take up all that disgusting but fantastic phosphate, amino acids, whatever they need, and they just don't care. So before I go, I'm just going to finish up. I'd like to thank not only the people that were on Team Hagfish, which are incredibly important to me, and some fantastic long-term friends that we've had. All this work, or almost all this work, has happened at Bamfield because obviously we have access to both hagfish and freshwater, but we also have access to great fishing, right? Which is important. I make good friends, and it's fantastic. And that's one of the things that we want to have. We did for a while have hagfish that we did some work here with the BSAS aquatic facilities, and I could thank Clarence and Tad and Marisha. At Bamfield, we've had a few researchers, coordinators, Bruce Cameron, Eric Clellan, and Tal Eastman over the last 15 years or so, who have been incredibly helpful in getting us going. And we wouldn't be able to work out at Bamfield and the challenges that are out there without fantastic help that is supplied by Bamfield. And I thank the Dean of Science for the idea that they continue to fund and support Bamfield. And I encourage anybody who wants to think about going out to Bamfield, even if you don't have a project out there, you can dig it up, and 15 years later, you might have 30 to 40 papers. You never know, right? So thank you very much. This is a cartoon that's in ACTA's editorial by Katie Gilmore. And with that, I'll take some questions about this charismatically ugly, but fantastic animal.
Great, thank you very much, Greg. That was fantastic. So we have to monitor the Zoom to see if there are any questions online. If there are any questions here as well. So I'll start at the back.
Greg, I'm wondering if there's if they slime themselves when they get in there, and there's any protection from the slime.
There is a suggestion that by some that there is some slime, but slime is very expensive to make. Each filament looks, so they have these intermediate filaments. They actually, Doug Fudge and I have done some work together, but he is the main leader of this. I just did some physiology. But there's a really amazing paper that came out that what happens is they make these intermediate filaments that are up to 18 centimeters long inside each cell. Centimeters, not microns, not millimeters. They're long, intertwined fibers. It's like a rope, like 2 or 3 filaments that wind like a braid on your hair. Right, and then it starts at the nucleus, and the nucleus spins and forms it like a ball of yarn, right? And it sort of loops, and if you're doing rope and you have to do the loops, same thing for these things. It loops, and then they have this in this tight hygroscopic component. And then when they release it, you need mucin, and it immediately within seconds unravels. So it's a whole secretion and unravels, and you get this fantastic slime formed within literally seconds. there is a suggestion, but you don't see it that often. You don't see slime unless you handle it. You touch them with gloves, they don't even slime. So I think there's an electrical sensation, but that's another thing that hasn't been really investigated. If you grab the animal, they'll slime, but otherwise, when they're feeding, they don't generally slime it. Some people thought that maybe it's kind of like your brother licking the food before, so you didn't get half of it. But that's, I don't think that that's necessarily true. So because we don't see it that often. Mark?
So tell me about ammonia in sharks.
Oh, so sharks are great because they take ammonia and they convert it to urea. And instead of doing ion regulation or ion conforming, like the same ion concentration, they keep their ion concentrations low. But now they can use urea as an osmolyte to maintain water balance. So they use urea and something called trimethylene oxide, a balance between those two, and that raises their urea concentration. That sort of replaces those ions in osmolytic sort of capacity. Right, so they have a very active urea production mechanism, very strong. So they use a different strategy entirely, sharks and rays. Stephanie?
Thanks, Greg. You mentioned that, they go in and they don't care about the conditions, but is there a point at which they do? Are foraging belts one long continuous exposure to these harsh conditions, or are there limits temporally that how does it connect back to sort of the energetics and other conditions that they're experiencing?
I guess we have to, I don't think anybody's ever done the experiment
Okay.
and tracked an animal going in or out
Really?
or whatever. Yeah, I don't think that's ever happened. We did do a study where we looked at using a wine maze and use different potential scent attractants. And they appear to be sensing ammonia and from animals that are decaying. So if there's a current, they'll just stream, they'll actually detect it and go against that gradient to be able to detect it. Or alternatively, propionic acid, butyric acid. So there's some suggestions. So, but no one's ever looked at whether they go in and out. They just know that they go in, and you don't really want to hang around for long when you got a putrefying dead animal. So no one's ever really looked at when they come out. We don't know anything. We know nothing about them. We have no idea what their fecundity. But their fecundity is low. They only have 24 eggs sometime every few years, maybe. There's a huge fishery for them. They wipe them out or semi-wipe them out in all sorts of places and then go to another place that has them. I have video of in Ucluelet, multiple 45-gallon drums of live hagfish just being dumped into a trawler for shipment to Asia. So it's an interesting idea. I don't know. There is a suggestion that for small fish, they're actually, because they can get rid of ammonia and some gases across their skin as well. So they might stick their head in where it's and then when the blood circulatory goes up to the other end, it excretes out from the other end. So there, but again, we don't really have all that information.
So you presume that being embedded in your meal also is protection from getting eaten by something else that's attractive.
Yeah, especially if it's as toxic as it is.
A trade-off in terms of vulnerability to yourself.
Today, has the microbiome been identified for this?
So I'm not working on this. Carol Bucking, who's at York University, she is probably working on microbiomes. I know she's the one who's really heavily into hagfish in there. I know there's some microbiome work going on with sharks with Alyssa Weinrod, who's my former PhD student. She's now at University of Manitoba. I don't know if she's doing anything with hagfish yet, but we actually show that hagfish can acidify their stomach, which is another one that's unique, that because they don't have a stomach, they just have a long intestine. They don't have a stomach, they don't have, it's really just a tube. It's a weird situation. They also don't really have stress hormones. They don't get stressed. Like you can put them in and just do as much as you want to them and they just hang out, they don't care. And there's no blood cortisol, there's nothing. It just is, they don't experience stress as we would consider stress.
Why is it such an unusual adaptation? You sort of painted it as if like there's these big dead things in the bottom of the ocean and nobody else can eat them. Why?
Well, they eat them after everything sort of floats away. When the animal blows up and blows, of course, yeah. Right, but while that putrefaction, while that carcass is still covered, there's an opportunity and they can get in there and acquire those nutrients as fast as can. It's a race, just like everywhere. You know.
It's a
they-
very temporally specific strategy as well.
Yeah, well, yeah, because they're opportunistic feeders, like they, opportunistic necrophagers, they will be on as soon as they can and try and get as much as they can before all the other animals get there to try and eat. But, you know, they might be months without a fall anywhere close to them. Or even longer. We could take them and put them in zero oxygen for 48 hours and they don't care. Their metabolism doesn't matter. Strange, but true.
How do they fish for them? How do you catch them?
There's hagfish traps, they're like tubes that are this long, they have small little holes, so that you put dead fish inside and they just crawl in and then they just wanna hang there because they think it's the inside of a putrefying animal and they just hang out inside there and you just lift it up. Banffield's great because Banffield's one of the few places that the shallowest that you can catch hagfish in the world, those shallowest report. Typically, they're at like 500 to 1,000 meters. They're all over, they're cosmopolitan all over the world except for the Antarctic Ocean. There's 73 different species, but Banfield is unique because literally half a kilometer offshore, you can drop a trap and get hundreds. And that's why it's a fantastic place to do the work. And I would encourage anybody to go out there, and if you need advice on how to do it, feel free to ask.
So if the RH protein levels are staying the same, is it just transport of the apical RH to the basal membrane, or?
I don't think it's to the basal membrane, it's going into intracellular vesicles so that it can be shuttled back into the apicus. Because each different transporter has its own targeting sequence, whether it goes to the apical or basolateral surface or whether it stays in a Golgi or wherever it stays, they have those targeting sequences. So molecularly, we don't know what those are yet, but that's essentially how those different RH proteins are assigned to different membranes.
So it's sort of like capping and then endocytose or something?
Yeah,
like
they
that?
just there's endocyte, it's very well known for a lot of different transporters that they can be rapidly endocytosed, especially in neurobiology, right? It's very quick, but we don't, this one's a lot slower. But metabolically, they're kind of slow. Any questions online?
There was one that, so yeah, we were having some trouble with the Zoom, that's why I went to see that, there was one, but it was exactly what Stephanie asked, so
Okay.
then they said, well, that's.
Wonderful.
So we should probably end it there. Greg, thank you very much for a fantastic talk and.
I appreciate it.
I'd like to offer you, very good, actually.
Great.
What about-
Haven't got one yet.
You can do what about-
Thank you, everybody. I appreciate you coming out.